and welcome to another episode of Max Planck's Neurotransmissions. I'm Lesley, your host for today, and I'm really excited to welcome our guest, Dr. Jeong Kim. Dr. Kim is a professor in the departments of Physiology and Biomedical Sciences, as well as the director of the Neuroscience Research Center and the director of the Memory Network Medical Research Center at Seoul National University, College of Medicine.
Yeah.
You sound like you must be really busy, so thank you so much for taking the time to come and talk to us today on the podcast. So you were actually trained as an MD, correct, at
Yes.
Seoul National University. Did you ever actually practice medicine clinically?
Oh, like 2 weeks
Oh.
after become a doctor, because my father he passed away 2 years ago, was a physician. So I just 2 weeks practiced in my father's clinic, and then I became a PhD student. So after that, only science and research.
That's right. So you did your PhD with Dr. Jeong Kim, where you studied the physiology of neuropathic pain.
Mhm.
What was it that made you make that transition from MD to PhD?
Oh yeah. So I actually like to practice also, but I thought at that time clinical practice is pretty much repeatable of the same way to people get better. That's very important job. But I thought at the time research is more interesting because every day is not like every day. I have to pursue new thinking and new hypotheses and new experiment. I thought that they might be much fun, but I didn't know there could be also a lot of failures. So anyway, I'm enjoying my decision at the time, and I really happy to keep learning and pursue my curiosity by doing experiment. That's really a good decision, I think.
That's great.
Yeah.
And you started out actually doing something that was probably quite relevant to your medical training, which was studying the physiology of neuropathic pain. Maybe you can just start by explaining what actually is neuropathic pain.
Yes. Pain is our perception, and you cannot imagine life without pain because pain protects yourself to be damaged from injuries, or you can to use your joint too much, you feel pain. So it really protective. That's we call as a good pain. But sometimes pains are not so helpful. So one example is neuropathic pain. When you get injured, usually and hopefully you will get better and back to normal. But some injury, like a nerve injury, does not get back to normal. It stays to the different level of sensory perception. So that we call neuropathic. Nerve damage induce permanent pain, which need medical intervention. But like aspirin or Tylenol, they can kill your pain. But that drugs does not kill your neuropathic pain because mechanism is totally different, which we don't understand. So we need to understand the mechanism of neuropathic pain to manage that inevitable, uncurable condition. So that's a clinically very, very important topic. And then, so I think that's why I'm focusing on neuropathic pain.
Yeah, it's definitely, I would think most people who have ever suffered from any sort of chronic pain, a topic that we need all the research we can get. And you've spent the last, I'd say, 25 years or so studying this topic. But what I found actually really interesting is that in parallel to studying neuropathic pain and physiology of pain, you've also spent a lot of your career studying the mechanisms of plasticity and learning in the brain. And these sort of two seemingly different topics, I actually really enjoyed your talk this morning, and we'll get more into that in a little bit, how actually these two seemingly different topics sort of united in your current research. But before we get to that, after you did your PhD work, you then went to do your postdoc work to study, sort of change gears a little bit and study molecular mechanisms of plasticity and even metaplasticity, focused on an important area of the brain, the cerebellum, with David Linden at Johns Hopkins University. And I'm curious, at the time when you were studying neuropathic pain, what actually led you to sort of change gears a little bit and travel around the world, and also to study a slightly different topic?
Yeah. So for the pain study, I was recording spinal cord at that time before going to Johns Hopkins. I was thinking that for the understanding of pain, I would like to go more higher brain network. I don't say spinal cord is not important for the pain understanding, but I'm more curious about higher level of brain as change in pain or not, and if there is a change in higher brain, how is it like? So I can choose brain areas between cerebellum or cerebrum, which is more highly organized network. But this all like some relationship, human relationship. So I have a very good friend who already went to the Johns Hopkins, and he get me to be connected to Johns Hopkins professor. Actually, he is Paul Worley, Dr. Paul Worley. So I contacted him, and then and he said we will do project together with David Linden, and then it's about cerebellum, and then I think it's wonderful because cerebellum is more higher than spinal cord, and then I decided to go to the cerebellar physiology, and also for the understanding of pain, for example, chronic pain, is not that much different in the sense of plasticity. So pain is another kind of plastic change of brain. So the understanding of very up-to-date study of cerebellar plasticity should be very related to understanding other plasticity like chronic pain. That's what I thought. That's I took the airplane and bring all my family to Baltimore, and I have had a wonderful 2 years at Johns Hopkins. So I really appreciate Professor Paul Worley and Professor David Linden, who is still very good teacher of my science career.
So I really appreciate how you actually draw these parallels between pain and plasticity. And, we know that cerebellum, as you mentioned, was sort of probably at least in my opinion, the most well-studied system that we have for understanding plasticity and even more complex concepts like metaplasticity. And that's actually something that you went into in your postdoctoral work, correct? Well, maybe you can give us just a brief overview of what is metaplasticity first, and then maybe some of the mechanisms that you found for this unique metaplasticity, which I was really interested to hear about in the cerebellum.
All right. Yeah, I would say metaplasticity is I would say plasticity of plasticity. So I would say in other way, sometimes teachers also can be more clever, or sometimes maybe in bad condition. So plasticity needs some good teacher. So it's like to learn, you need some mechanism to be learned. So when he's saying metaplasticity, it's the plasticity of that mechanism which induce plasticity. Could be plastic. It's a kind of more higher level understanding of learning and memory. So any molecule which detect the experience and then which can induce plasticity could be a target of metaplasticity. For example, today's my talk, I mentioned metabotropic glutamate receptor, which is very important molecule in the plasticity mechanism. And this metabotropic glutamate receptor detect the experience of neurons because it has a glutamate sensor, and also it has a its activity. Its activity depends on intracellular calcium concentration. So also its downstream is protein kinase C. That's also activity dependent, also calcium dependent. So it really senses the environment of the experience, and it can eventually change the synaptic connectivity strength. So it can teach the synaptic strength. So the change in metabotropic glutamate receptor, if it really function more stronger, that could induce next event more efficiently be plasticity. So that's a concept of metaplasticity. Then we really excited when we found that that teacher could be plastic in the cerebellar system. There's a very good study in hippocampus, and that study usually focus on NMDA receptor, which is also coincidence detector of the activity. In the case of cerebellum, Purkinje cell, the amygdala was doing like NMDA receptors. And then we, actually it was excellent finding though, but we could find that amygdala signaling could be different at some event, and it could be more lowered. And then this lower level of amygdala metabotropic glutamate receptor, so I usually say amygdala, but it's a metabotropic glutamate receptor. And then it really changed next coming event experience. So when metabotropic glutamate receptor is reduced, the next time induction of plasticity also reduced. So it really changed the plasticity. That's what I have found and reported.
Great. So it's a way of really tuning how easily our synapses can actually
Definitely.
learn.
Yeah. But that specific molecule depends on the location of the brain, I think. Yeah.
So there are a lot of different plasticity mechanisms throughout the brain, and even ones that may be involved in pain.
Oh, yeah.
Right?
Yeah.
So in your talk today, you actually had a really interesting, you talked about some preliminary results that were really interesting where you actually were looking at the mechanism of pain perception in the cerebellum. And we don't actually normally think too much about pain and the cerebellum as sort of fitting together, at least for me, who's not a cerebellum expert. I normally think of the cerebellum as involving, movement control or maybe error prediction or, of course, plasticity. But this morning you were saying, no, we should really be paying attention also about the role of cerebellum in pain. And how was it that you first actually thought about considering the cerebellum as a site of processing pain information?
Yeah. Um, so it's kind of a personal interest at the beginning because I was trained as a pain physiologist as a PhD student and become a doctor of science. Then I had a very good experience, as I told before, as a cerebral physiologist. And while doing that research in my research career, so I really saw that cerebellum is really related to the area where I found a pain physiology related. So my curiosity began. There is a good connection from cerebellum to the other brain areas, which is related to pain processing. Then what's the role of that connection? And I think there must be some function of that connection. And I'm trying to find the first time as a research, I trying to find what pathway conveys that pain information to the cerebellum. So that's the beginning of my talk today. I found unknown pain input pathway to the cerebellum and try to find the function of that input.
And what was that specific input?
Yeah, there's some brainstem area, locus coeruleus. So that locus coeruleus is activated by pain input to your skin. So if you activate pain afferent sensors to the skin, the locus coeruleus is activated, and then that locus coeruleus send axons all over the brain. So when I'm thinking as a textbook wise, its output looks like a brain-wide activity. It includes cerebellum also, and then we try to record that activity specifically in the cerebellum, and we found that pain stimulation to the skin also activate the terminal in the cerebral cortex. That means that's one of the pathway convey pain information to the cerebellum.
So you saw that, the cerebellum was in fact, getting some information about pain sensation in the body. So I was really interested in the finding that you did next, which was what was downstream of that. And you found these Bergman glial cells in the cerebellum to be the target of that locus coeruleus input. And, again, I was surprised. Bergman glial cells are these, I know them because they're just these gorgeous, if you stay in the cerebellum, which of course is such a beautiful region, right? You see these beautiful Bergman glial with their, long processes extending out to the surface. But, I hadn't really thought too much previously about sort of their integration in the cerebellum circuit and what they might be doing, particularly in the context of pain. So how did you discover that these cells were actually involved and what their, putative role might be?
Yeah. So I'm not the first researcher. Bergman glial is very active. So Bergman glial is one of the astrocytes in the brain, specifically located in the cerebellum. So it really, as a glial, it supports neuron circuit. But what I really surprised is that that Bergman glial also excite when locus coeruleus input is activated. So Bergman glial has no adrenergic receptors, And then lococellular neuron is the most input, which release noradrenaline in the cerebral cortex. So we could hypothesize that Pokemon glia is one of the target cell when lococellular is activated by noxious pain stimulation in the periphery. So we decide to record both type of cells at the same time, and then we successfully recorded that when we give pain, the lococellular terminal activate in the cerebral cortex, together with Bergman glia, calcium also increased. So that's a very good finding. I can suggest that Bergman glia is the first new cell in the cerebral cortex, which process the noxious information in the cerebral cortex.
Oh, neat. So, they're processing this information, but are they actually regulating the animal's perception or response to pain? How do you even measure pain in a rat? You can't really ask the rat as we would a human, like, is this painful?
Yeah, I'd like to ask.
Make it easier.
But I need some language with mice. But yeah, you are right. So that's why we use behavioral analysis of animal when we cannot talk about their condition. So that's right. So we found a very good activity of cells in cerebral cortex, but whether it is really contributing to pain perception or pain behavior, we don't know. So we need to observe whether that cell is really cause that kind of perception. So we need kind of a animal behavioral model to understand and then to figure it out. So we used capsaicin induced pain model. So as you know, capsaicin is the component of hot pepper. So when you taste it's hot. Also, it's a very specific agonist of C fiber in the peripheral neurons. So we injected capsaicin in the paw of the mice, and then how do you feel? You feel some hurt on your foot. You may scrub those foot until you feel better. So mouse is doing same thing. When you feel pain in the foot pad, mouse licks the foot pad, and then how much they lick is how much they feel pain. That's how we interpret the amount of pain perception of the mice without asking how much you feel pain. Instead of we observe behavior. And then the Bergman glia is the main pathway in the cerebellum is supported by behaviors. How we did is we controlled the Bergman glia activity by using chemogenetic manipulation. And then that chemogenetic manipulation suppressed Bergman glia activity in response to pain. Also, together we suppressed the leaking time of mice also. So it means the Bergman glia activities related to the pain processing in the cerebellum.
Wow. So not only is it detecting the pain, but it's actually modulating the pain.
We suggest so. Yeah.
Interesting. Now there are a lot of other brain regions, of course, and of course the spinal cord that are involved in pain perception and pain behaviors. How do you think the cerebellum sort of generally fits into sort of the overall processing?
Yeah, that's still a question we are trying to answer. So we need a scientific evidence about that. But we know that as a circuit wise, not functional circuit, is anatomical circuit wise, cerebellum is very much connected to pain processing other brain areas. For example, cerebellum is a very good closed loop circuit with prefrontal cortex, and that prefrontal cortex is pretty much related to pain, cognitive pain perception, and also cerebellum is also connected to periaqueductal gray, which is the main system of the endogenous analgesic system, which means it really controls the pain threshold of the spinal cord. And then if cerebellum can modulate any of these, I'm just saying, I'm not saying all the list of possible possibility, but I'm just saying example of the cerebellar possible connectivity related to pain.
Sure.
Yeah, so that's kind of experiment ongoing in my lab. You'll see about which is the main target or how cerebellum really modulate pain perception or behavior.
Great.
Yeah.
Well, we look forward to seeing that
Yeah,
for sure.
thank you.
And I know you have a fairly large group of scientists working with you, and you've had a lot of really successful scientists actually move and train through your lab. And so I thought maybe I would just ask you if you had any particular advice maybe to a younger scientist who's maybe starting out their PhD, and just starting their career.
Yeah. I'm so happy man who did good science in my lab. I really appreciate it. So my advice is choose good lab and then read a lot. And don't be disappointed because most of your experiment could be failure, but we learn from error like a cerebellum. So I think there's always a good chance of fail, also a good chance of success. So don't give up by the failures. Just keep going and move science, and then that could be very fruitful eventually.
What great advice.
Yeah, yeah. Read a lot. Read a lot is important.
I always like to hear about sort of the latest exciting results from your lab, and I'm wondering if there's anything new or something that you're most excited about in your lab right now that you would like to share.
Oh yeah. Let's see. Yeah, we are the group where we reported plasticity of intrinsic excitability. So people generally think there's a synaptic plasticity, but also there is also plasticity of. I mean, synaptic plasticity means plasticity of synaptic molecules like AMPA receptors or NMDR or mGluR. But there's also ionic channels on the dendrite or soma. That is also plastic. So we are the group where we reported that membrane ion channels are also plastic. And then we are looking for what's the function of the intrinsic excitability plasticity. That's one project we are so up to and excited.
Really exciting. Well, I look forward to reading more about that work in the future.
Yes.
Great. Well, thank you so much for joining us on the podcast today. It's been wonderful chatting with you.
I enjoyed a lot. Thank you very much.
This has been a production by the Max Planck Florida Institute for Neuroscience. You can listen in on iTunes or SoundCloud, follow us on Facebook or on Twitter @NeuroPodcast.